Ever pondered how the invisible becomes visible? How the intricate world of cells, hidden from the naked eye, is unveiled? Welcome to the realm of cell biology and microscopy, where we'll delve into the techniques used to bring the unseen to life. Step 1 on our journey brings us to microscopy, a scientific technique that allows us to see objects and structures that are too small to be seen by the naked eye. It's essentially a superpower, enabling us to peer into the minuscule world of cells and their components. To understand microscopy, we first need to grasp two key concepts: magnification and resolution. Magnification is the process of making something appear larger than it is. Imagine looking at a tiny ant through a magnifying glass. It seems larger, right? That's magnification at work. Resolution, on the other hand, is the ability to distinguish two close objects as separate entities. Think of it as the sharpness or clarity of an image. If you've ever tried to read small print and found it blurry, you've experienced a lack of resolution. Now let's move on to the two main types of microscopes used in cell biology: the light microscope and the electron microscope. The light microscope, also known as an optical microscope, uses light to pass through a specimen and then through two lenses to create an image. It's like a superpowered version of the magnifying glass we mentioned earlier. These microscopes can magnify up to 2,000 times, but their resolution is limited to around 200 nanometers. That's because light has a certain wavelength and it can't resolve objects smaller than that. On the other hand, the electron microscope uses a beam of electrons instead of light to create an image. These electrons have much shorter wavelengths than light, allowing for a much higher resolution. Electron microscopes come in two main types: the transmission electron microscope and the scanning electron microscope. The transmission electron microscope, or TEM, shoots electrons through a very thin specimen. It can magnify objects up to 1,000,000 times their actual size with a resolution of less than 1 nanometer. The scanning electron microscope, or SEM, scans a focused electron beam over a surface to create an image. SEMs can magnify up to 500,000 times with a resolution of about 2 nanometers. In a nutshell, we've journeyed through the fascinating world of cell biology and microscopy. We've learned about the principles of microscopy, magnification, and resolution. We've discovered the capabilities of the light microscope and the electron microscope, including their types and their magnifying and resolving powers. So the next time you see a close-up image of a cell, you'll know the science that made it possible. But remember, this is just the tip of the iceberg. The world of cell biology is vast and ever-evolving, so keep exploring, keep questioning, and keep learning.